All right. So, any of the proteins that participate in this replication are referred to as the DNA replication machine, or it's basically the replication complex. So as I said, this particular model, the hypothesis that is supported, is one where DNA polymerase molecules actually reel in parental DNA and extrude newly made daughter DNA molecules. So again, reeling it in, tugging it through it, rather than actually moving down. Yes, Richard.
You said that both DNA polymerase 1 and 3 are working at the same time.
Yes.
If they're both pulling on it, then how does that work out?
DNA polymerase 3 pulls on it. DNA polymerase 1 could also pull on it, but if you look back here, because all the polymerases are moving in the same direction, or would be moving in theory in the same direction on a single strand, that's a moot point. If this polymerase here were pulling on it in this direction, and this polymerase here were pulling it on the other direction, that would put stress on the molecule. But because they're all pulling on it together in the same direction, it's going to work probably more like people in a tug of war would work. If all of them heave together, and then heave together, and continuously pull the molecule through.
Okay.
Okay? Okay, so finally I want to talk about mismatch repair, because Richard did bring up the idea that there could be some mismatches that occur while you're doing this, and you're not wrong. Okay? There can be times when polymerase is not perfect. You know, nobody's perfect, so neither is polymerase. So sometimes it's going to mismatch an A to a C, or an A to a G, or G to a T, or a C to a T, or even a T to a T. And when that happens, we actually need to excise or remove that mismatched nucleotide. So what I'm saying basically is, if the parental strand is AAA, you know, maybe our polymerase gets the first two right, but it could then potentially put an A or a C or a G for that final one. We don't want that there, because we want this to be maintained, because in this other strand down here, it was originally TTT. And we want it to be the way it was before. We don't want it to be TTA, we want it to be TTT. So this is where we're going to utilize enzymes called nucleases. And what nucleases actively do is they can actually excise nucleotides. At least a particular type of nuclease is capable of doing this. So what it's going to do is it's going to move in where the mismatch is. It's going to excise that particular piece of DNA, or those nucleotides. Then a DNA polymerase is going to come in, replace those nucleotides, and DNA ligase will once again come in and ligate the DNA together. Okay, so this is what happens in the event of mismatches. So, there's one last point I want to kind of make about this whole process. In our cells, on our DNA, because it is linear, not circular, like bacterial chromosomes, we're going to encounter some problems when we go to replicate. What actually is going to happen when we go to replicate here is we're going to wind up with these ends that our DNA polymerase is actually not capable of completing. In other words, at the 5' ends where we have primers, we're not going to actually be able to supplement that particular region of the DNA with more DNA, or complementary DNA. Which means every time we replicate this strand of DNA, we're going to start to make shorter and shorter and shorter pieces of DNA. Okay, so if you look right here, when we get to the end here, we've got this primer here that basically goes away once we're done replicating, but we don't replace it with any DNA. So then, when we go through the second round of replication, the new leading strand is going to actually wind up being shorter than the parental leading strand. Okay? Questions, concerns about this? This is the principle behind aging. These structures that exist at the end of the linear molecules of DNA are called telomeres. And every time your cells perform replication and they divide, the telomeres in your cells and in your nucleus get shorter and shorter and shorter. And this is why.
How does that relate to aging?
How does that relate to aging? Well, if you have a gene here on this DNA, let's say this is a gene that encodes some protein that is required for your survival. Every time your cells divide, this DNA is getting shorter and shorter and shorter and shorter, and eventually you're going to get to the point where with another division, you run the risk of cutting off a piece of that gene. So your cells actually can undergo one of two processes. They can undergo what's known as apoptosis, or programmed cell death, or they can undergo another process that's called senescence. Senescence is simply a state where the cell can no longer divide, but it is still technically viable and living. And that's technically what aging is related to. Basically, when you age and you get to a certain age, what's really happening is more and more of your cells are incapable of dividing and replenishing themselves if damage is done to the body or if a certain stress causes them to rupture or burst. Yes, Stephanie?
Is that like when the pigment stops?
Similar to that, exactly. So that also has to do with the idea of stem cells no longer being totipotent and actually just becoming regular cells that have differentiated and no longer can divide an unlimited number of times. But yes, eventually you can get to a point where you no longer produce pigment, and part of the reason for that is a lot of the cells that maybe were producing that pigment no longer are dividing as much. At least they're not dividing in large enough numbers to make enough pigment to give you brown hair. Yes?
I've heard of experiments to try to reverse the process, try to increase the length of telomeres, and I hear that they actually don't do any good.
Well, here's an interesting fact. First and foremost, telomeres are those structures. This is actually what I got my Nature paper on, was telomeres. And these particular structures in telomeres that add to their stability. Okay? So these don't prevent the shortening, but they do prevent or postpone the erosion of genes. And they seem to have structures that also help them be shortened in a very regulated sort of way. Okay. And just to show you a picture real quick, these are all chromosomes. I'll turn the lights out real quick because I think this is kind of cool. This is fluorescent in situ hybridization, which is a fancy way of saying we've got fluorescent markers that actually attach to particular parts of these chromosomes. Okay, so these are chromosomes, and anywhere you see the yellow, those are telomeres. All right, now, going back to what Richard said, yes, there have been attempts to actually add in telomerase. In fact, your germline cells that give rise to your gametes, your sperm and your eggs, those actually do have telomerase expressed. Okay, because those cells typically are maintained for a very, very long time, and that prevents sterility. But the rest of your cells in your body typically don't have telomerase expressed. Telomerase being the enzyme responsible for lengthening telomeres. Now, mice actually have large quantities of telomerase expressed in their cells, and mouse cells have longer telomeres than human being cells do. Anybody know the average age or life expectancy of a mouse, though?
3 years?
About 2 to 3 years tops compared to ours. So having telomerase isn't always a good thing. And what's even better, most cancer cells seem to have telomerase activated. So this is right now one of those holy grails that scientists are constantly seeking, saying if we can just find a way to manipulate telomerase, we can extend human life. Because ideally what that would then do is allow your cells to divide an endless number of times. But guess what other cells can divide an endless number of times?
Cancer.
Cancer. So we might actually try and make something that helps us, and it could wind up backfiring. Okay? So yes, as it says here, telomerase activity present in cancer cells. Say that again, Lorraine? The telomeres? I'm sorry, I still can't hear. It's okay. Here, I'll come closer. Right. So sometimes it can be based upon actual telomere links, presence of telomerase. I haven't done extensive research in turtles, so sadly I don't know. But it also could deal with, if you look at turtles though, their metabolism, very, very, very slow. Okay? Because they're reptiles for one, so they're going to actually, their blood and the way that their body works is going to be slightly different from the way ours does. Whereas mice and most mammals, much higher metabolism because we're warm-blooded organisms, so that might also contribute to it. That would be interesting to look at. Okay. So, looking at DNA in humans or in mammalian cells or animal cells compared to, say, prokaryotes. First off, eukaryotes have linear DNA in their chromosomes. So we have this linear DNA that's wrapped around proteins to give us our chromosomes. Bacteria, on the other hand, have these circular chromosomes. And these circular chromosomes are associated with a small amount of protein, and it's called the nucleoid. It's typically supercoiled. Okay, and of course, remember, bacteria don't have a nucleus, so it's going to be found just in the center of the bacteria, not in any kind of membrane-bound envelope. In our cells, we're going to have DNA wrapped around these histone proteins to make this structure that's known as chromatin. Okay? And here's how we're actually able to stuff so much material into such a teeny tiny space. The double helix of DNA is wrapped around these histone proteins to make this chromatin. Okay? So we've got this bead on a string, and you'll notice we make multiple beads. What we're then going to do is we're going to actually perform what's known as supercoiling, where we then take these beads and we wrap them around each other to make this larger structure. Okay? There, that larger structure of chromatin, where all these beads are wrapped around each other. And then we're going to take that larger structure and we're going to wrap it around itself. Okay, into these loops and scaffolds that will eventually make up the fibers that will make up our chromosomes. If you were actually to take one of these chromosomes and stretch it out, it would be incredibly long. In fact, what's that, Jeremy? What's... Yeah, there's enough in each and every one of us here to go to the moon and back probably several times, if I had to guess, and this is why. Okay? Because what we're actually doing is taking a really long string, and we're wrapping it around things to make it tighter and more coiled, and we're wrapping those things around themselves to make them tighter and more coiled, and then we're going to do the exact same thing again. Yes?
I noticed that the name histone, that's similar to histamine. Is there any connection there?
I imagine there is. I haven't done extensive research on the primary histone compared to that of histamine, but I imagine that there probably is some, at least some relation between that and histamine. So. Okay, so now, in the last 10 minutes, I want to start on what the final lecture is going to be over, which is transcription and translation. Just to let y'all know, I know this chapter is ridiculously long. We're not going to make it through the whole chapter. I will tell you what will and won't be on the exam based upon where we get. But I do want to, at the very least, make it through transcription, RNA processing, and hopefully translation between today and Thursday. Okay? So, what we're going to talk about in this chapter is the concept of gene expression. That is, how do we take the message in our DNA and convert it into a polypeptide